Welcome to the fourth seminar of our fall term in our Ecology, Evolution, and Conservation Biology series. Before we begin, I'd like to thank the Departments of Forest Ecosystem and Society, Fisheries and Wildlife, Integrative Biology, as well as the Forest Biodiversity Research Network and the Oregon State University Research Office. Special thanks to Amanda Pauley for running the technical aspects of Zoom and YouTube today. And finally, a thanks and a welcome to our speaker, Dr. Wendy Turner. Wendy earned her PhD from the University of California, Berkeley, studying the ecology of gastrointestinal parasites in herbivorous mammals in Namibia. And Dr. Turner has been conducting research in Southern Africa her whole career. Dr. Turner is currently a research biologist with USGS, an assistant unit leader of the Wisconsin Cooperative Wildlife Research Unit, and a senior visiting scientist at the Department of Forest and Wildlife Ecology at UW-Madison. And recently, she's been working on a range of topics, including, of course, anthrax transmission and pathogen evolution, but also the fungal disease white nose syndrome and chronic wasting disease. And with that, I'll turn it over to Wendy Turner.
Okay, thank you very much. So I will start us off today with a quotation, actually from the plant pathology literature, but I find it very inspiring for my own work. So one of the principal challenges in epidemiological analysis is to understand the causes of the variability that occurs between different epidemics of the same disease. And I find this to be a really great quote that encapsulates the kind of work that I'm trying to do, because in a lot of disease systems, depending on the characteristics at a certain time or in a certain location, outbreaks can be really small, or as we've been experiencing this year with COVID, they can be very big. And trying to figure out what are the driving factors for this is something that I'm working on in my own research. And so there's a lot of different and very interesting examples for emerging infectious diseases, showing us really interesting examples of how quickly diseases can evolve in their transmissibility and in their severity. We've seen that in viral biocontrol in rabbits, and then in various wildlife systems, when a new emerging disease comes in, that it can change and become more or less infectious and more or less severe to the population. Although there are numerous examples of this in emerging infectious diseases in naive populations, there's also examples in sort of ancient diseases or old friends of ours that have really fascinating variation in the transmission dynamics. So new research out this year on the plague bacterium in China shows that depending on the climate conditions, the pathogen can evolve different traits to enhance its transmissibility. So when it's cold and dry and the vector populations are quite low, the flea numbers are low, it can evolve traits that improve its chances of transmitting from the flea to the mammalian host. So in my own research, there's sort of two driving goals of my program. So the first one is to determine the causes and the consequences of the spatiotemporal variation in wildlife disease outbreaks, why it varies in one location at different times across a disease system. And then secondly, because we're working in wildlife systems, it can be really hard to understand what's driving transmission. You know, we talk an awful lot about this year publicly talking about things like R0. And it's really hard to get at that in a wildlife system where you can't actually do experimental work. It's hard to manipulate the system. So we do a lot of work of, doing really intensive data collection, looking at the ecology and the behavior of hosts, digging into the genomics of the pathogen, and then trying to pull all of that together with statistical and mathematical models to try to get at all these things that we can't just do experimentally. So in my lab, we've been working on primarily on anthrax. In the last couple of years, in my position in New York, started working on blood parasites in songbirds and white nose syndrome in bats. And in my new position, I'm starting now in Wisconsin, I'm going to be working on chronic wasting disease in white-tailed deer. But today we're going to talk about anthrax, and you really can't talk about, well, any disease system, especially anthrax, without bringing in the epidemiologic triangle. So this is where you have the three components of a system. You need to understand the host and the pathogen and the environment, and the interaction between the three of those component parts is where you get a disease outbreak and transmission happening. So the approach that I take is to try to understand transmission by focusing on all three of these aspects and looking at how the host pathogen contact occurs and how that's modulated by the environment, how the environment affects both survival of the pathogen, because most of the work I do is on environmentally transmitted pathogens, so those that have some portion of its life cycle, if not a very long portion of it, in the external environment, and then how that environment modulates the susceptibility of the host species. So anthrax is the disease we're going to talk about today. So anthrax is a highly lethal bacterial zoonosis. So it's a pathogen that infects both wildlife, livestock, and humans. And the really interesting thing about anthrax is that it is globally distributed. But depending on where you get an outbreak, it varies in its seasonality. In some locations, you see wet season outbreaks. In other locations, you see dry season outbreaks. The incidence can be quite different, from a few sporadic cases to really explosive outbreaks. Which species are affected can vary from place to place. And the outbreak interval can be quite different. So you can see annual outbreaks occurring each year, and you can see locations like a recent outbreak in reindeer in Siberia, where you see cases that you haven't seen them in 50 years. And I find all of this really fascinating. So if you think about a typical anthrax host, anthrax can affect any mammalian host, but it's primarily herbivorous hosts. So you do get the occasional case in a carnivore, but it's mostly the herbivores that are being infected and dying. But these herbivores are wildly different in their ecology, in what they're eating, how they use the environment, from hippos in pools to your cervids, your bovids. It's a very interesting disease system because it has a very broad host breadth. And so if you think about the life cycle of Bacillus anthracis, it has two life stages. So you have the spores, which is what we think about when we think about bioterrorism, because these spores are very hardy. They can persist for years, decades longer, depending on the conditions. And then once those dormant spores get into a host, they turn into the vegetative cells, which are the growth form. And so the question is, when you have these spores out in the environment, how do they then get into a host? So you have a couple different routes of transmission. So you can have ingestion, so generally thought to be through grazing, browsing, or through drinking. It can be through inhalation, and it can also come cutaneously through biting flies. And this figure I'm showing you here has been adapted from the WHO's most recent guide to anthrax. And the text here is directly copied from that figure because I think it's informative. How little we know about the transmission, is there inhalation or not? How is it ingested? It's quite surprising to me that an important disease like anthrax has so many questions about how it's transmitted. And then once it gets into the host, it rapidly kills the host, generally within a few days to a week for most herbivores. And the interesting, I suppose, Achilles heel for anthrax is that the lethal doses are really, really high, which you generally want. If you have a highly lethal disease, you want it to not be especially lethal unless you get a very high dose. So depending on the route of transmission, those doses can be different. So if you think about an LD50, which is the dose that will kill 50% of your population, the lethal doses, the LD50s for inhalational anthrax, are much lower than they are for ingestion. So inhaling it, it can kill you more easily than it can if you consume it. So from experimental studies, we know that inhalational exposures are on the order of 10 to the 4 to 10 to the 5 spores, whereas for ingestion, it's on the order of 10 to the 8. So 100 million spores is thought to be lethal. And so knowing what we do about transmission, which is, well, I guess not a whole heck of a lot, I got really interested in studying this in an endemic system in Southern Africa. So I've been working in Etosha National Park in Namibia, which is a large national park that has a very characteristic salt pan, which you can see here. It covers about 1/5 of the area. It's a very flat park. There's very little topography. And all along the edges of that salt pan are grasslands. And in these grasslands, sort of south and west of the pan, are where we see a lot of anthrax cases. These are just some typical pictures of what this location looks like. And our key 2 host species that we see, well, more than 50% of cases we record are zebras, and we see the next most important host in the system would be the blue wildebeest. But if you look across sort of the host community, we see these are the top 5 host species, but most of the cases that we see, most of the confirmed cases are zebras. And then when you get down to the oryx, the gemsbok, only 1% of the cases that we record, and then all other species are only 2% of cases detected. So really most of what we see is in these 5 species. So if you think about the fact that whether there's ingestion or inhalation, I'm going to take you through 3 different studies that look at host pathogen contact through these 3 routes of transmission. For this, I'm focusing on grazing because our 2 main host species in this system are both grazers. So the question is, given that we know the lethal doses are extremely high for this disease, the place you're most likely to get that high of a dose is going to be at a carcass site where an animal died and is opened up by scavengers, and less likely to be where a scavenger transmits spores through feces or bathing at a water hole. So I'm going to start with the grazing pathogen, or the grazing transmission pathway. And this work was really kickstarted by me spending way too much time in the field during my PhD. And there was a big outbreak of anthrax in 2006. And this was me coming back the next year and finding one of the carcasses that I had recorded as an anthrax carcass and seeing this, the grass growing here. And so this is early in the wet season, and you can see this flush of vegetation here amongst the bones. And it really got me wondering about how a carcass would influence the grazing cycle of animals, and whether you would have avoidance of a site like this or attraction to it, depending on whether you're more drawn to the grasses or repelled by the carcass remains. And either way, that's going to really affect the transmission dynamics if you have either attraction or repulsion from these infectious sites. So this became the foundation of my postdoc work, was working at carcass sites to try to understand what's happening to these locations. So I asked a series of questions. Firstly, do nutrient pulses from carcasses improve local soil and grass quality? Because basically you're just dumping a bag of fertilizer into the environment. Secondly, can herbivores be exposed to Bacillus anthracis if they graze at these carcass sites? So we have plenty of studies have shown if this is a soil-borne pathogen, you can find it in soil for years. Do animals have to have contact with the soil, or can they get it through grazing? So the picture I'm showing you here is a grass on the Etosha Plains after a rainfall event, and you can see all along the blades of the grass are soil particles. So how much potential is there for exposure simply through grazing at these sites? And then finally, how do animals behave around these carcass sites? Are they attracted to them for the vegetation, or are they repelled by the remains of the carcass? And what's the time scale over which that those differences occur? So this is going to be years of work thrown into a single slide here. So I did a lot of sampling of plants, the soil, the bacterium, and the herbivore behavior at carcass sites. So this is what a typical carcass site looks like in Etosha. And this is on the order of a couple days after death. Scavengers are very happy in Etosha. They consume quickly all the soft tissue. The bones get drug off. You can see out here in the back is the remains of the skeleton. But these sites get really quickly consumed by scavengers, and you can see this sort of denuded area of vegetation where you have bare soil with the sort of the liquid contents of the carcass, the gut contents and the blood go into the soil along with the pathogen. Everything gets stirred up by scavenging activity, and then that's kind of a clean slate, if you will, waiting for future changes. Over time, you get regrowth of vegetation at that site after rainfall events. And you can see you end up with greener, higher biomass grasses growing at those carcass sites. They tend to be of higher nutritional content. And through sampling of soil and vegetation through time at these sites, also looking at, sorry, cut off a bunch of slides here, also looking at the pathogens, so looking at the concentrations of the bacterium on the blades of the grass, on the roots of the grass, and then in the soil surrounding these plants, and how that changes over time. And then finally, using camera traps with a carcass control, case control design to look at how animals are using these sites over time, and using these white rocks here, which are found in the environment, to give depth of field to the photos so we can see that they are in the site and how they're behaving. So this was a 3-year study, and I'm just going to summarize the results here for you. So for these 5 species studied, we basically found that these were all zebra carcass sites, and elephant and the oryx were hardly ever detected at these sites. If they visited them, they never foraged there. So their risk at these sites was very low. For the other 3 species, the 3 perhaps more important species in the system, we see that they do show attraction to these sites. So in terms of the nutrients, we see increased forage quality and biomass in that first year after death, but it doesn't really persist beyond that. We see that the spore concentrations are pretty high on the grasses 1 to 2 years after death. And then thereafter, it's still present in pretty high quantities in the soil, but we don't see it moving onto the grass as much. In that sort of early window of time, that first 1 to 2 years after death, we see that these carcass sites are attractive to some of the species, but not to others. And of the 3 who did show a significant attraction to foraging at these sites, the zebra, the wildebeest, and the springbok, zebra were significantly more attracted to others. And I believe they were 4 times as likely to forage at a carcass site as they were to forage at a control site nearby. Okay, so that gives us some confidence that grazing is a potential pathway in this system. And then how about in relation to its companion transmission route, if you will, the other ingestion route through drinking? And when it comes to disease management, this is a very important pathway to consider, because if you have an outbreak in a location and you have a lot of scavengers in the area, things like vultures, they like to feed and then come to a nearby watering point and they bathe their feathers. So there's been concern about the risk of scavengers moving the spores around and potentially concentrating them at water holes. This is particularly in locations that have point source water, which is what Etosha has in entirety. There's no river systems there. So to compare these 2 pathways is a bit of a challenge, because we know, okay, they're both coming in through the ingestion route, but we don't know much about dose-response relationships. So how do we know how much contact you're going to have with the bacterium at a carcass site versus in a watering hole? So for this work, I paired with a colleague, Wolfgang Bayer, who had been working with the graduate student in the park, looking at watering points. So between the 2 of us, over a 5-year period, I had been doing work looking at a bunch of carcass sites and sampling them each year to look at concentrations of the pathogen, where he and his graduate student at the same time had been sampling the sediments of water holes of different types and trying to look for any seasonal patterns as they dry up, as they become wet, and how that affects transmission. So if I just throw up here the data from combining these two data sets, we have a log 10 scale here, and we have everything in rainbow colors is my carcass site data, and everything in pastels over here is the water hole data. So you can tell right away that you're seeing much higher concentrations of the pathogen at a carcass site than what you see transferred by scavengers to potential watering sources. Of what you see of the carcass sites, the higher sampling here, these are the soils, the surface soil at the carcass site over time, whereas these other samples here are all grasses, either the above ground biomass, the below ground biomass, or the soil surrounding the roots. So we see that that surface soil is obviously the highest, but there is decay over time. And then if you think about transmission in this system, so when I showed you earlier in this talk, the lethal doses for this pathogen, when it comes to ingestion, the expectation is that 10 to the 8 should be about 100% lethal. And so there's been a tendency in this system, or in this disease system, to say, okay, well, 10 to the 8 should be lethal, but most of what you sample in the environment is many orders of magnitude lower than that. I got a few cases up here, a few soil samples early on that crossed that threshold, and this is all log per colony forming units per gram. So this is scaled to the mass of either the soil or the vegetation. But it's fairly easy to look at this and say, okay, well, we know what should be lethal. We know what we're recording in the environment, and it's so different. Why is this? And the explanation has always been, okay, well, these studies are done in laboratory animals. You know, these are wildlife out in nature. They have other infections. They might be more susceptible, and it's probably just that. But I don't find that very satisfying because you don't know exactly how much contact an animal is going to have with grasses, with soil, with water. So we put together some models to try to understand what the risk would be. So I'm going to first show you some plots here looking at carcass site data. And this is just focused on potential exposure to the pathogen through foraging on grasses, that above ground biomass component. And so this is a somewhat complicated looking graph, but we have a 3D surface here. On this axis here, we have the probability that what you contact is going to kill you. So from 0, it's not going to kill you, to 1, it's definitely going to kill you. Then we have an estimate of grass intake, and we assumed, based on average bite size of a zebra, that between 0 and 30 bites of grass you might have at a carcass site. And then in terms of the lethal dose, this is the unknown in the system. We don't really know what's going to kill an animal in the system. So we just estimated it. We have from 0 out to 10 to the 10. And we know from experimental studies that 10 to the 8 approaches 100% lethal. And so this is looking at pretty much the sites initially. So 0 years since death of the host, what's that initial soil concentrations at these carcass sites. You can see that under these conditions, pretty much any foraging that you do is going to quickly jump you up here into pretty high risk levels. And as time goes by at these sites, those risk levels are going down, but they still retain somewhere around 10 to the 5, 10 to the 6, out to 4 years later, you see a jump that your risk is not 0. So as you get, assuming that you can get a lethal exposure around 10 to the 5, 10 to the 6 spores, there certainly is potential even 4 years out after death, which was the length of this study. How does that compare to what you see at watering holes? So the water hole data is a little bit more complicated to work with because what my colleague and his student are measuring is the sediments under these water sources and how much the pathogen gets stirred up and can make it up into the surface of the water to be consumed. It's going to vary based on a whole bunch of factors that we don't have data for. in terms of how many animals go into the water, who's been there before, was an elephant taking a bath, etc. So there's a lot of complexity to that. So we just did a simple assumption, and I'm only going to show you one of these, but if we assume that in this graph here, 1/1000th of what exists in the sediment has the potential to get up into the water column to be consumed, what does that exposure look like? So again, we have this probability that's lethal from no to absolutely. On this axis, we have water intake, and I put this on the log scale because we went out to what a really thirsty adult male elephant might consume on a hot day. So this is a huge amount of water consumed. And again, with the uncertainty in what actually would represent a lethal dose. And what we see is that basically, even for elephants consuming a huge amount of water, it approaches zero. So it's moving down towards singleton spores are what would be required to kill you based on what's possible in the water. So in terms of your risk, the risk of contacting the pathogen at a carcass site is much higher than it is at a water source. So if we come back to our contacts here, the next one we wanted to look at was inhalation. And so the inhalation study was kind of twofold motivated. So on one hand, we don't quite know what's happening with elephants. They're not being exposed to these carcass sites, but elephants do this dust bathing behavior, which is very dramatic. And if you think about the potential that if there are spores in this soil that they're bathing in, they have a pretty high potential for exposure to the pathogen. And the other thing is that elephants tend to die in the dry season, and they take more dust baths then. So it seems like this is a important potential pathway for this species. The other thing that motivated this study was that we had a zebra that died of anthrax right on top of a dust bath. And we thought, oh my gosh, this is like totally a super spreader event just waiting to happen. So we put a camera up on it right away. And this became the foundation of Zoe Barandango's master's thesis. And so she did a survey of dust bath soils and quantified how much of the pathogen we found, and she did observations of animals dust bathing at different sites. And what she found ultimately was that of all these sites that she surveyed, she only found spores on 2 of them, and in very low concentrations. It was 10 and 20 colony forming units per gram. And if you think about for an experimental inhalational anthrax study, and these are done with pure spore suspensions put inside an animal's nasal passages, in those examples, your lethal dose is on the order of 10 to the 4, 10 to the 5 spores. So we did some back of the envelope calculations to estimate how much soil you would need to inhale at the levels that we detected it in the environment, and found that these animals would need to inhale on the order of 1 to 10 kilograms of soil to get a lethal dose through this route. And quite frankly, if they are inhaling that much of the pathogen or that much soil, it's not going to be anthrax that kills them. So this makes it seem that this pathway is not all that important in the system. And the question is then why? So we had a bunch of carcass sites that we've been monitoring for years in the system. And we found that these are examples of carcass sites 2 years after death. And you see that they have this sort of persistent, what we call a gut cement in the environment. So when you have the scavengers consuming the carcass and compacting that soil with all the contents of the gut, you get these patches of really hard soil. And I think that what happens is that these soils are not that, they're not soft, they don't easily break up. And so an animal is gonna, start dust bathing somewhere, but probably not at a carcass site. So just to sort of summarize the transmission component of this study. So in terms of grazing-based exposure, that does a fairly good job of explaining what the 3 top species in our system, the zebra, the wildebeest, and the springbok. Thus far, we haven't really been able to pin down how elephant and gemsbok are getting exposed in this system. another one of our master's students from the University of Namibia, Hendrina Joel, she's been working on elephant carcass sites to understand, do elephants have specific behaviors that affect their exposure at these sites? And from her own video cameras on these elephant sites, she's found that both elephants and the oryx have pretty high, relative to the other species, exposure to the bones. So the elephants will come and sniff and pick up the bones, and the oryx will come and actually chew on the bones, probably for micronutrients. So it seems like there's potentially a different pathway if you look at a different host species. So one of the things that we've been working with in this system is to try to understand how much of a dose of the pathogen an animal is going to get through these different behaviors. So we went through the different routes of contact, but then we were wondering, can we estimate some of these infection parameters through other means? So with my colleague Ryan Easterday, we sort of wondered whether we could look at the genetic diversity of the pathogen at death and use that to estimate back in time how much, so whatever you ingest into the GI tract, only a portion of that is going to cross the epithelium to infect the animal. And then you're going to have proliferation across the duration of the infection. And then after death, sporulation occurs, and then there's going to be decay over time. So we were wondering, if you looked at the genetic diversity of the pathogen at death, can you estimate that initial population size and the time from infection to death? So for this work, we sampled 11 zebras, and we isolated 30 colonies of Bacillus anthracis from each one of these carcasses, and did some genetic work to look at their diversity. And in tandem with that, we did an in vitro mutation rate experiment. So we took a single isolate of the pathogen, and we replicated it into 60 replicates, and we ran those through 6 different transfers to get an estimate of the number of the genetic diversity of these 60 samples at the end of the experiment, plus the number of generations that occurred during that experiment. And so we used coalescent theory to put these 2 different data sets together. So this is a model of how gene variants that are sampled from a population have originated from a common ancestor. So you can use, if you look at the time of death, looking at the genetic variation there, you can trace back in time based on a known mutation rate to the timing of infection. So with this, we were able to estimate that in this population, the infection duration or the time to most recent common ancestor was on the order of 1 to 3 days, which seems blazingly fast, but it really matches what you see in the literature. And then in terms of the infecting population size, it was a little bit harder to pin that down because we started that mutation rate experiment with a colony, which is several hundred cells or more. So we were able to get it down to an estimate of 1 to 3 colony forming units, which we think is probably on the order of several hundred cells. So really you might need a huge exposure to have enough cells that can cross over the epithelium and start the infection. But the starting population size seems like it's fairly small and the infection's really fast. So the work that I've described thus far has been really focused on the details of transmission, what's happening at individual sites, what's happening within animals. But my goal all along was to try to look at sort of bigger picture questions of how animal movements on landscapes affects the transmission dynamics. And our ongoing work that we've been doing now is starting to focus on this. So now that we know enough about the transmission process to really understand how and where animals are exposed, then we can start to look at the bigger picture questions to understand why we see seasonality in the system and why we see such differences from one year to the next in terms of the size of an outbreak. So this is work I'm going to show you now of, it's currently unpublished, but hopefully soon, my PhD student Yanhua Huang, looking in this at zebra movement ecology, comparing wet years versus dry years, because we, by chance, had collars on animals during the worst drought on record. So if you think about spatial patterns in anthrax mortalities, I'm going to show you what we see in the system. So I showed you those grasslands next to the pan are where we see most of the cases. If you look at a simple vegetation map, you can see that where we see the most of the cases happens to be more open landscapes. So do we just detect them more because there's more roads there for more tourists, and it's easier to see things in a grassland than it is in a woodland? This has been a question that's really hounded us for a while in the system, is how much of this is a real ecological pattern of that particular environment, and how much of it is just that we can't have equal surveillance across the park. So one way to get at this is we looked at the ratio of non-anthrax to anthrax mortalities that have been recorded through time in different groups of species. So we know that in Etosha, we tend to see many more cases in grazing host species than in browsing host species. So we wanted to compare the two of those. So what you see here is this is just a sum of mortalities detected over time for grazing hosts versus browsing hosts, from open habitats to shrubland habitats to woodland habitats. And what you can see is that, well, obviously we detect a lot more anthrax carcasses in open habitats, but in comparison to the non-anthrax mortality causes, things like predation, we see much more anthrax in the open areas. For the browsing hosts, we do detect more cases in woodlands where we expect them to spend more time, but we don't see that there's a real uptick in anthrax risk. So this, in some ways, supports what we see in terms of browser grazer risk, and it provides some evidence that what we're seeing, there could be an element of surveillance bias, but there does seem to be a real pattern that there is higher risk in those locations. So we wanted to compare animal host use to look at seasonal and interannual patterns in outbreaks. So we did this with GPS collared zebras. And we had two study periods. In 2009 and 2010, we had a pretty average rainfall year, but it happened to be the largest anthrax outbreak on record, or not on record, since the 1960s, close to largest ever. And then that occurs in the wet season. So we have here in the blue, here is just all the locations from these collared animals, and you see them really clustered in that plains habitat where we see a lot of cases. In terms of dry season locations, they spent still quite a lot of time there, even into the dry season. When we look at our drought year in comparison, in the wet season and the dry season, you see animals are not spending quite as much time on those grasslands. They're sort of pulled back into the woodlands further away from the pan in both seasons. So it seems like the way they're using the habitats is differing in the 2 years. If we think about their resource selection, what proportion of time they're spending in these different habitats, you can see how different their use of the landscape is between these two periods. So in our large outbreak year, most of the fixes, more than half of them, are in those open plains. And then the sum together of the grasslands and shrublands is most of their time spent. In our drought year, we see most of the fixes, on the order of 70% of what we're recording, they're spending in the woodlands. And if you just take a simple picture of that sort of open habitat in these 2 years, it's not terribly surprising that they're not using this location. So we see in that drought year, there's really nothing to consume on those plains. So again, it's not surprising that animals aren't using that. But then, that's partly perhaps why we're not seeing as many cases. If there's something about that plains habitat or how animals use that habitat that's leading to more cases, If you have years like a drought where animals are not there, even if they are much more susceptible, we're not seeing the cases at that time. So in terms of our next steps, where are we going from here? So we have a pretty good understanding of how the system works in Namibia, in this grazing system. So we have expanded our study to look across 2 different ecosystems in Southern Africa, one that's dominated by grazers, Etosha, and one that's dominated by browsers in Kruger National Park in South Africa. And for this study, this is an NSF grant, their EEID program. We have a really big collaborative group that's doing this work with PIs, including myself, Pauline Kamath at the University of Maine, Henriette van Heerden at the University of Pretoria, and John Mfune at the University of Namibia. And together with our collaborators in the national parks in both of these countries, we're trying to dig into the underlying factors that make disease outbreaks in these 2 systems quite different. So if we look, let me introduce you to Kruger National Park. So Kruger is a similarly sized park. It's also a savanna ecosystem. It has very similar very similar animal species there. But the differences are that this is a much richer environment. You have these major river systems that cut east to west across the park, which can inhibit animal movement. And when we do see anthrax outbreaks up there, they tend to be in the far north of the park, and they can sometimes spill down further south, depending on the size of that outbreak. But our main host species in this system are typically kudu, and then in more recent years, there's been an increase in impala cases. In this system, the outbreaks have been tied to a mechanical vector, blowflies, leading to an enhanced transmission. So you have your initial carcass in the environment, and we're not exactly sure how that happens. That carcass gets fed upon by blowflies, which have this massive population explosion. And you can see in this photo where the vegetation all around is just completely covered with blowflies. These flies are regurgitating and defecating spores of the pathogen onto the leaves of trees and shrubs. They don't like to land on grasses. And so when you have a browser such as kudu in the environment, the potential for exposure at these locations is pretty high. So you think about comparing these 2 systems. In some ways, it seems like a very simple grazing versus browsing comparison, but it's a bit more interesting than that. I guess you'll say complexity is interesting in my mind. So in Etosha, we see relatively small outbreaks that happen annually. In Kruger, we see a large outbreak interval, so on the order of a decade between outbreaks. And when they do occur, they're really big outbreaks. And if you look at the top 5 host species in each system, well, you see some repeats. We see zebra and elephant occurring in both systems, but our main host species are either the grazers or the browsers. And the nice thing about these systems, so the ones I've circled in or highlighted in orange here are only found in a single system, and the rest of these host species are found in both systems, whether or not they're actually an active anthrax host or not. And if you think about their feeding type, whether they're a grazer, a browser, or a mixed feeder, it's more complicated than just the one system is grazing based and the other system is browsing based, because the only species that's actually a pure browser is really the kudu. So moving forward, coming back to that epidemiologic triangle, trying to understand the 3 components of this system and how that affects your contact, your susceptibility, and your survival becomes complex and fun. So we're looking at the genetic diversity and the plasticity of the phenotype for the pathogen, as well as the genetic diversity of the host. We're looking at all sorts of aspects of the environment and how that differs. We're looking at how that environment-host interaction is changing in each system. All the different factors in terms of the soil characteristics, the soil microbiome, bacteriophages, how that can all affect how well the pathogen survives in the environment, and then how all that comes together to affect your transmission pathways and how animals are interacting with that environment. So it's just a couple interesting points about these two systems and a few interesting, it's all very interesting, some preliminary results from this work. So what you can see is that anthrax is a seasonal disease, but that even in a single system, we can have different types of seasonality. So this is data from Etosha, where we see the main peak with our zebras. These are in % mortality per species, so the dry season ones are blown up a bit here. But there's two different peaks driven by different species. And the same thing is true in Kruger. We have a dry season peak and we have a wet season peak. And one species is either contributing to one peak or the other. One of the things that we're trying to get at is not just the individual movement ecology, some of the habitat selection that I showed you for the zebra, but comparing it for the two main host species in both systems, and then layering that in with camera trap grids that show us how the population density of these species and the whole community is going to change over time, and how that's going to affect your risk, your number of potential hosts on the landscape at a time. And a few more preliminary results is that these systems, even though they have the same complement of host species, are really different. So in Etosha, the home range sizes are huge. They're several orders of magnitude higher than they are in Kruger. And they're even, even for the same species, kudu and Etosha have a much bigger home range than kudu and Kruger. And this is probably in part due to the productivity of the system and the rivers that inhibit movement in that system. And if we revisit this epidemiologic triangle, you think about it in terms of a generic disease system, you have sort of equal components of host pathogen in the environment. If you think about this in terms of anthrax and how we tend to think of this disease, it looks a little bit more like this. So it's really driven by the environment. You have the right kinds of soils and that semi-arid environment. There's an element of host. You need to have a herbivore there, but it's not as important as that environment that's going to promote spore survival. And in terms of the pathogen, we don't have much expectation that the pathogen is going to contribute much to these systems. And why is that? Because anthrax globally is considered a monomorphic pathogen. It's wherever you find it, it's very similar. This is work postdoc in my lab, Spencer Bruce, was doing, looking at the global genomic diversity of this pathogen. And really, you can see from the shallow tips here, there's not that much genetic differentiation across this pathogen. So this sort of expectation that you have then is that you have a clonal pathogen in the system. If you have variation between ecosystems, it's probably going to be driven by the environment and the host and their relationship, and not necessarily by the pathogen. However, it isn't necessarily the case. So what we see in Etosha is really what you would expect to see for a clonal pathogen. That this is just the genetic diversity of the pathogen that we see in the park, and then the relative frequency of these different strains through time. And you can see what you see is most prevalent in the 90s, or in the 1980s, excuse me, is prevalent through time. So that sort of matches what you'd expect to see if the pathogen doesn't necessarily have much variation in the system. If you look at data from Kruger, though, that kind of makes things more complex. So Kruger has a lot more genetic diversity of the pathogen than what Etosha has. And you can see from one decade to the next, there's almost complete turnover of the strains that are prevalent in the system. And then the question is, why? You can ask me questions about that later. And then just a final note, one of my other PhD students, Zoe, she did master's with me, and then she came to the US to do her PhD as well. She's been looking at trying to link this pathogen genotype to phenotype, because there's hypotheses out there. There's certain clades of this bacterium that don't persist as well in the environment. So we've been looking to see why is that, looking at characterizing the diversity, looking for things like phages in the genome or in the soil that are going to affect the phenotype of what we see. And I just had to show you one last result, just because I think a fun thing about the kind of wildlife field research that I do is that no matter how much you think you understand a system, there's always something that can come along and surprise you. So we've been doing long-term studies of zebra anthrax carcass sites. We've been sampling for, I think, 11 years now. And we understand what's happening at these sites in so many ways. And then Zoe, just last week or so, showed me some data looking at the initial spore concentrations found in soil at these carcass sites based on the season in which the animal died. So if you were a zebra and you died of anthrax in the wet season, which is our sort of anthrax peak season, you had several orders of magnitude more spores in the soil than you did if that animal died in the dry season, when we don't typically see many cases. And this just really blew my mind. And then I started, is this affected by the weather conditions? Is that seasonality driving sporulation success in the few days after death? Or is this seasonality through host susceptibility? Are animals in the dry season more susceptible to dying of anthrax and so die with a lower terminal cell count? We don't know the answer to that yet, but hopefully we'll in the future. That is just a nice way to end this talk. Got lots of acknowledgements for the many people through the years who have helped me with this work, and the many people who are now doing this work instead of me while I sit in the office. And that's it, and I thank you very much for your attention.
Okay, Seb, do you want to take it away?
Yeah.
Please submit questions to the, I'm sorry, please submit questions to the question and answer tab. Thank you.
I think, yeah, we have a few more minutes. And one of the first questions we have is from Dr. E, who asks, is it known why scavengers are not affected by anthrax?
No. I mean, in terms of what is it about a scavenger that makes them not susceptible when the herbivores are, we don't really know. Work that's been done in the system looking at serology in scavengers shows that pretty much as soon as they start eating meat, they have sky-high titers. So they might have better protection than herbivores who are exposed to lower doses. There's also an argument that animals that feed on carcasses have generally a stronger immune system, especially around their gut, and that maybe that's why they are less likely to die of the infection, but we don't know.
Okay, we have another question from Marie, and she asks, are there any pathogens that could predispose animals to contracting anthrax? And makes the point that rarely are animals exposed to just one pathogen. And is there any evidence for synergy between pathogens and environmental conditions that would make exposure more possible?
Absolutely. There's been quite a lot of work from a colleague of mine. We were both PhD students at the same time looking at co-infections with nematodes, with ticks, and how that can influence their risk of contracting the infection or their risk of, or their susceptibility. Because the argument is that animals should be most susceptible to a pathogen like anthrax in the dry season when they're resource limited than in the wet season when they're not. So Carrie Szyszowskis was my colleague doing that work. And that's certainly something to consider. We have another student in South Africa, Sunday Okai, who's looking at that now. So he's looking at bloodborne parasites and finding some really interesting interactions between the bloodborne parasites and the immune response against anthrax.
Thanks, Wendy. Kristen asks if you could expand more on why there is turnover in anthrax strains in Kruger versus Etosha.
I don't know the answer to that yet. My hypothesis is that the river systems have something to do with that. So you have these major river systems that cut through the park. The locations in the north there where you tend to see outbreaks are on floodplains. So my wondering is if you're having something wash downstream from somewhere else and that you're getting turnover over time in what's coming into the park. That it isn't necessarily a closed environment.
Okay, so we have a question from Anna who asks, okay, so what is up with higher genetic diversity of anthrax and Kruger?
I don't know the answer to that yet, Anna. I'm working on it. But again, the assumption is that it's probably linked to the river systems, that you have this more potential for stuff coming into the park, and it's not just the diversity that's there within, like what we expect from Etosha.
Rosario asks, how is climate change expected to affect future outbreaks? Will more animals die during the longer and more extreme dry seasons, but will soils around these carcasses may have lower levels of viruses? Which do you think will be a more important driver?
That is a great question, and we don't quite know. I mean, Southern Africa is expected to get hit quite hard with climate change, with increasing temperatures and increasing desertification. And when you have most of these park systems are fenced, so animals can't sort of move into more suitable climates. So we expect an increase in stress on these animals, whether that translates into increased susceptibility, how that influences sporulation of the pathogen in the environment. So we do see that in this drought year where these animals were severely stressed and nutrient limited, we saw hardly any anthrax cases. Whereas in a really high rainfall year when there's resources galore, we see tons of cases. So it's not a direct correlation that drier, more stressed animals equals more anthrax. One of my PhD students, Amelie Dolphy, who's just getting started, is going to be looking at questions like this, trying to build models of the system and look at it under different climate scenarios and things like that, how that's going to affect the transmission dynamics.
Okay, I think we have time for one more question. And this is from Spa NR. Has your group considered the effects of artificial water provision on game numbers and the effects of the spatial distribution and transmission of anthrax in Etosha National Park as Kruger National Park is currently doing? This is clearly somebody who knows what's going on.
I know. Well, so Kruger National Park had this sort of addition of all these artificial watering points up the park system, which has influenced which species are there, and it sort of brought the zebra up. And there has been some change in the Kruger system over time. So from the 1950s through the early 1990s, when we saw outbreaks, they were basically only kudu was the main host. In the past 10 years, there's been a shift in the system that we're seeing it more in impala, which are about a 50-50 grazer browser, and we're seeing more zebra cases. So the question is there something about that, that we brought the grazers to the north through the water provisioning? Did that also sort of shift the system in terms of how frequently we're seeing those outbreaks and which species are the ones being affected? In terms of Etosha, we basically only have point water sources. So there's both natural springs, but there's also these artificial watering points. Those artificial watering points used to be throughout that grassland habitat where we see a lot of anthrax. Until in the 1960s, they had several years of massive outbreaks in that location. So as a result of that, they actually turned off all the artificial watering points in that habitat, forcing animals, because it used to be a migratory system. They used to migrate all the way around the pan each year. Fence line cuts the pan off to the north, and provisioning water there year round enabled them to stay on the plains. So by cutting out that water, they started migrating again, at least back and forth around the edge of the pan, if not circling it. So long story short, yes, I think that there's certainly the interaction between water, how that affects the density of hosts available in the environment, certainly plays in with the vegetation and how that influences how much exposure they have when they're foraging.
All right, thanks again for coming to our seminar. Next week we have another speaker, and I'll share that information here in a second. And oh, can you see my screen now?
Yes.
Okay. So thanks again to Wendy Turner for an excellent talk. And please join us again next week, the same time and the same place.
Thank you.
Thanks, Sebastian.
Yep. No problem.
Great talk and good turnout again. So
Do I need to hand this off to somebody else here?
no, I think we're pretty much done. It looks like Wendy signed out. So
Okay.
Yes, I was hoping to chat with her a little bit.
how's it going, Tal? No, that was not a loud cough. It was just a medium cough. Oh, he's gone. All right, I'm off. See you guys.
All right.
Bye. I'm going to shut it down.
Bye. Thanks, Amanda.
No problem. You did great.